Okay. So I will just say a few words about the midterm, but you will get marks like today afternoon because I finished marking. Marisa just gave me two last quizzes, two last midterms that we have to pick up from ACS. But everything else I marked. And we will go in details. Marisa will probably talk more about that next week because I'm out. Uh, Overall, really good. Like the one that when I marked it first, like 3 quarters, the average was around 80%. So pretty good. Like that's really, really good. And I'm counting everyone, not just 311, like 311 and 511. Grad students, absolutely amazing. Like that's average like 90, which is great. That's what we expect. I didn't expect anything less. But also undergrads, I can tell you that the highest mark is 98.5, and that's in undergrad. Yeah, so somebody got 98.5% in undergrad, beating grad students in that. So some new nerds, new children are coming, guys. What to tell you? You will. It's hard field out there. So no, and there are many like really reading your written responses. I was usually I get very annoyed when somebody says something exclusively incorrect, but that didn't happen this time. Like not at all. Like everyone is fantastic. Like not everybody, nobody said anything exclusively incorrect. So it's really good. I want to think that I inspired you to think like that. I don't think that's really correct, but let me live in my own dreams for a while. Uh, because microbial physiology can be really fun once you fall in love with that. I know it's hard to love it. That's like it's high maintenance, but once when you fall in love, you stay stuck for the rest of your life. Same like me with this amazing TCA cycle that I want to talk about today because that's what that's where we left off last week. Um, TCA cycle is my favorite metabolic pathway, and I don't think I ever knew something better than that. And I just because it's going everywhere, like it's going through microbial physiology, it's going through animal physiology, plant physiology, and even with slight modifications in the side roads, TCA cycle is always there. So, and there are key enzymes that I will talk about. So, how, again, to go back to this stuff. So we talked about how pyruvate in fermentation can go to lactate to ethanol, and the purpose of having lactate dehydrogenase and ethanol dehydrogenase is to just uh, regenerate that oxidant, like that NAD plus, so the cycle can operate, and that's all related to glycolysis. And we constantly talk about that NADH, NAD recovery, etc., etc. Now, this is all nice and good when we are strictly talking about anaerobic metabolism. What happens is when you have aerobic metabolism, then you have involved electron transport chain or ETC. And in order to get ETC operating and to get that final ATP production that happens at the end, what you have to have something to start that ETC, and it starts with NADHs and it feeds up to the FADH. So where those guys come, they come from TCA cycle. So let's go back to the product of glycolysis, which is pyruvate. So pyruvate is the product of glycolysis. If we talk about aerobic metabolism, is going to be converted to acetyl coenzyme A by an enzyme called pyruvate dehydrogenase. And as you can see, during pyruvate dehydrogenase reaction, NAD plus is reduced to NADH, which means that pyruvate is oxidized to acetyl coenzyme A. Acetyl coenzyme A is a key molecule that gets sent into the TCA cycle during aerobic metabolism. And the way how it works, like if you think about cell and the membrane that surrounds the cell, TCA cycle, even though we showed this TCA cycle usually in the center of the cell, TCA cycle is actually occurring like think about these circles like on the surface of the membrane. They're just tied to the membrane. So you have multiple of these cycles like surrounding the membrane. Why membrane? Because that's where the electron transport chain is happening, and that's where this TCA cycle is going to be associated with. So now, how we get that point? So acetylcoenzyme A now was built from pyruvate via pyruvate dehydrogenase. And acetylcoenzyme A, in order to enter the TCA cycle, needs to be condensed with oxaloacetate. Oxaloacetate is one of the metabolites that is a key metabolite in the TCA cycle, and it's significant for downstream pathways, which you will see later, because oxaloacetate is actually precursor for amino acid aspartate. So many of these TCA cycle metabolites will serve for the amino acid metabolism. So again, when I think of the end of oxaloacetate, we get this citrate or citric acid. Citric acid, in addition to having a lot of applications in applied microbiology, is a very significant molecule in TCA cycle. So citrate will be converted to aconitate or cis-aconitate, where you will have dehydration. That's where aconitase, like cis-aconitase, does. And then cis-aconitate is going to be converted to isocitrate. So again, you have aconitase. So I'm not going to ever ask you to know all of enzymes, but some of the key enzymes I do want you to know. So from aconitate by aconitate, you get isocitrate. So very often you will not even see this aconitate being shown on this cycle, simply because it's usually complicated for people who are trying to make a point. But the point is that from citrate, when you get isocitrate, somehow you have to get one of the most important metabolites in TCA cycle that I will ask you to know, which is alpha-ketoglutarate. Why I want you to know alpha-ketoglutarate? So it will become much more clear when we talk about nitrogen metabolism and glutamate, because alpha-ketoglutarate is going to be beneficial for glutamate. Now, again, that's a different story. Let's not focus much on it. One way to have isocitrate. What you have to get alpha-ketoglutarate, and the end of that is that dehydrogenase. Which dehydrogenase is isocitrate? Because it's made by the sucker that. So based on this energy running, what can you focus? What kind of reaction is it? In terms of energy, exit body for everyone. Because it produces, you have energy plus, so you have production of energy. So it's catabolic exergon. That probably the delta G is going to be minus, which means it is energy. If you can NADH oxidize it, because that is spending an energy between delta G and it's and it's anabolic. Again, those are questions that I think money solved in the quiz. So I'm trying to help. And I usually ask those exergonic and metagonic therapies that my son find out. Then I want to respond by knowing to recognize this. And whenever you see dehydrogenase, what that tells you is that there is oxygen reduction involved. So that means so in this reaction, NAD plus is reduced to NAD, while isocitrate is oxidized to alpha-ketoglutarate. So I never memorized all of that at the top of my head, but I do understand that if I want to see what is oxidized, what is reduced, I look for factors. Reduction, so that means 4 factor reduced, is oxidized. So I just have to oxidized. So that's what we will have. Now, alpha-ketoglutarate has a bunch of roles in organism. We will see when we talk next week. We won't be able to finish this week because nitrogen metabolism is quite complicated. Alpha-ketoglutarate can be aminated to glutamate, but that's downstream. If we stay on this cycle, alpha-ketoglutarate again is going to be what? Oxidized to acetylcoenzyme because alpha-ketoglutarate, and this is going to be done by alpha-ketoglutarate, the alpha-ketoglutarate dehydrogenase because this form. Now, What you get in this reaction is succinyl-coenzyme. Succinyl-coenzyme, this is the thing you have to be able to figure out. Succinyl-coenzyme is another reaction. And the enzyme that will catalyze this reaction is synthetase. What is making succinylate? This is what things get a little weird because the enzyme is called succinyl-coenzyme synthetase, and it's actually making succinylate. But I wouldn't like. I often make mistake and I say succinylate synthetase, or whether I explain that the enzyme is making succinylate from that. What you get is a reduction between. So again, that's about it. So the reaction synthesis sites are all exergonic and catabolic. Where does the GPP go? Downstream metabolism. Where you will use it? Protein translation. You will use it translation on ribose. Like everything what you make in TCA site has a use. Now, that's here is what? What is this one? How we call it? Is it quinol or quinol? This is quinol. That we use is quinol. Now, what this is reduction. So, as I said, everything is logical. You don't have to really memorize and you can easily conclude. Now, humans gets to be condenses with water, that's hydration, with and you get malate. Now, malate is subject to malate dehydrogenase, and already know the name. So dehydrogenase subject malate, you get reduction to NH3, you get what? Oxaloacetate. Oxaloacetate goes back and condenses with. Again, upcoming acetyl-coenzyme, which comes from pyruvate production into suicide. Now, where is the connection with TCA? Everywhere. NADHs, all of those TCA. One thing here that is not shown, which I don't like, but that's how picture is. Reaction of succinylate dehydrogenase is doing what? It's feeding into the complex 2 in the glycogen, which is very squirrel. So this is the same squirrel that we talked about when we talked about glycogen. We talked about the additional cofactor that gets reduced here is FADH. So FAD flux gets reduced to FADH. That succinylate dehydrogenase is part of complex 2. That's why this guy has to be close to the membrane, so it can associate with the complex 2. Complex 2, even though it's not proper fast, you don't get proper multi-force, it still contributes to the energy formation because of this FADH. And again, quinol is part of PTC and it's part of TCA. Does that make sense? Okay. And as usual, I give you a lot of varieties, but everything what I just said is written on these slides. Like I'm not going to read it. I just showed you the picture, but this is for you guys, so you can, if you want to read it, then you can go and read it. You don't have to go back and listen to my lecture if it's easier for you to just read. Now, what is the purpose of TCA cycle? The purpose of TCA cycle is, as I said, to generate reduction equivalents like materials as NADH and FSH for oxidative phosphorylation, and materials for biosynthesis. So the reason why I want you to know alpha-ketoglutarate is because alpha-ketoglutarate you will see all the time in metabolism of amino acid because it gets on in the most important methods of and later on glutamate gets second time amino. We will talk about that after I come back from Venice. Oxaloacetate is the molecule that always constantly needs to be replenished to get titrated because oxaloacetate condenses with acetylcoenzyme to get cycle acetic acid cycle. how you get another source of phospholipid. So that the phospholipid is going like what is this? So before you get pyruvate, you get phospholipid. And then the phosphate goes out and you get pyruvate. So that phospholipid doesn't only have to be used for production. It can be carboxylated to get oxaloacetate. You can keep going with the TCA cycle. What are products of TCA cycle? Most important, NADHs, FADHs, and ATP, GTP, carbon dioxide. So these are all useful compounds for our for bacterial metabolism, not our. Although and for our, but we love bacteria now. So what is glyoxyl cycle? So you probably now see the whole point of TCA cycle and how important it is for it. Glyoxyl cycle is the modification of TCA cycle. It's a version of TCA cycle. Not all microbes are using it. Some microbes have ability to use glyoxyl cycle, but not all. So, and again, if you look briefly, let's look like a TCA cycle. What's the difference? This breath. So what that means? So the reason why they do this, so like how they do it, they are catalyzing the addition of acetyl coenzyme A, but they are not producing these downstream products of TCA cycle. So the difference is that in glyoxyl cycle, so this cycle is not going to be converted downstream to the alpha ketoglutarate and the rest is the second coenzyme. So organisms that run glyoxyl cycles that can TCS and cyclic bias. So what this method does is break down the cyclic, the isocyclic, so it cuts it down, and then you get glyoxyl, that's what we call glyoxyl cycle. And so the key element of glyoxyl cycle, and we probably recognize the organisms running and another and then we skip a glyoxyl pathway which is now simply because glyoxyl is now equivalent to oxaloacetate in TCA cycle. It gets like condensed that's coenzyme goes out and glyoxyl plus glyoxyl plus malactate. Why do you have here mouth? Because mouth is obviously mouth hydrogen to get oxaloacetate. Because you have to get somehow so that's the purpose of the glyoxyl cycle. So which microbes use it? Microbes that live in environments that are rich with fatty acids. And these microbes that live in fatty acid rich area usually can use acetate as a carbon source. So it's usually very abundant, but not all microbes can use it as a carbon source. Only microbes that have isocyclic lyase and malate synthase can. And again, this is just a writing of everything what I just said. So you don't have to only listen to my lectures. You have everything in writing. I know some people find it useful, so I know I like to that myself. Now, since we are talking about electron transport chain and connections of EPC with electron transport chain, so you now probably can make connections all everything between EPC and everything downstream is transferred to electron transport chain. So and you now already know there are unusual electron transport chains with particles. But one of the important things, one of the interesting things that I wanted to share at the end of the lecture, this unit, lecture, unit 6, is that unusual electron transport chain that exists in between some of the microbes we got in our lab. And I found it interesting to understand the whole point of evolution and bioenergetics. It's ammonium acid method oxidation. And how can you see that? So what is the point? So again, very interesting. And I usually ask this as a bonus question on final exam because I think it's very hard. But if people can understand, then they get like a bonus points. And most questions usually replace a few marks for mistakes. So just a brief reminder. what is ammonia, what is methane oxidation, and who is doing it. So methane oxidation completed by methanotrophs. So they use methane or methanol as a sole carbon source. Again, we know methane is not contaminant, but greenhouse gas that harms our environment. So who are ammonia oxidizers? Besides the fact that they're very hard to grow and they really don't want you, no matter how much you love them, they just hate you. Very nasty oil. They can be very, very useful to us. It could be bad, but they could be also very useful. So ammonia oxidizers are known as nitrifiers, which means that they are performing the process of nitrification, which we will talk about more when we talk about biogeochemical cycle. But the ammonia oxidizers are using ammonia NH3 as an energy source. As a carbon source, they use carbon dioxide. So at start, I would say they have nothing common with methanol because methanol use methane as a carbonate and this source ammonia use ammonia as an energy, but carbon dioxide as a carbon source. So how are they connected? Why do we care about them? So you will see in a few minutes. So why are ammonia oxidizers so important? They are significant from an environmental perspective because they can oxidize ammonia to hydroxylamine, which is NH2OH, and then hydroxylamine gets oxidized to nitrite, NO2 minus, and nitrite or NO2 minus is later fed to another organism in an environment called nitrite oxidizing bacteria or NOB. But why is that important? Because sometimes when there is anaerobic environment or not anaerobic, but microaerophilic environment, which often happens in soils due to the loss of floods, global warming, ammonia oxidizers in soil become oxygen limited. When they are oxygen limited, this NO2 minus is not going to be sent to NOB, but they will switch their metabolism and they will do reduction of nitrites to nitric oxide, and then nitric oxide will be reduced to nitrous oxide. And nitrous oxide is a highly potent greenhouse gas, way more than that. So this is one of the connections, like they're related to global warming. So why do we talk about them? How are they actually connected? So methane, and I'm purposefully putting a formula here. CH4 and ammonia NH3, when it's in methanommonium or ammonia, depending on which form we talk about, are very similar. So both of these enzymes are metabolized or oxidized, like degraded by two very evolutionary similar enzymes, methyl monooxygenase in methanotrophs and ammonia monooxygenase in ammonia oxidizing or AOB. So MOB is methane oxidizing ammonia oxidizing bacteria. So those are technically same. If you look at them, they both need same cofactors, they both need copper, they are both embedded in members. But even though they look the same from evolutionary perspective, they still developed or evolved in two different routes, in two different organs. And those two different organs are ammonia oxidizers and methane oxidizers. So these are just comparations. So this is methane oxidation in methanotroph. So you go from methane to methanol. This is the beginning is that enzyme methane monooxygenase and then you go all the way down for the now. What's happening in ammonia oxidizing organisms? Again, I think you already heard about this, but in methane oxidizing organisms, you have two routes, ramp and serine cycle. So serine formaldehyde is the one distinguishing branch. So you have branch of formaldehyde assimilation in gamma methanotrophs, which is ramp. In alpha methanotrophs is the serine cycle. And the key product of serine cycle, acetyl coenzyme A, which can be shunt either to TCA or PHB or other cycle. So we will talk about this when we talk about applications of microbial physiology in different units. Now, methane monooxygenase or particulate methane monooxygenase or PMMO is using molecular oxygen to activate methane. And this particulate means it's embedded in membrane, so that's why it's called PMMO. There is another form that's called soluble methane monooxygenase. That one flux around it because it's iron as a cofactor, so it's not important for this purpose. So PMMO is using two electrons from anything that is electron donor, method, those two electrons from reductant. Reductant is NADH, and the electron donor, like whether it's like what gives, like is it a methane, is it NADH, like who gives those electrons? That was a big mystery, but it was literally solved a few years ago. Like who is donating those oxidation of methane? Is it a methane? Is it NADH? It's different actually in different metanotrophs, but most of the time it's NADH. So where is the difference between those organisms, like from physiological perspective? So the difference is in cytochromes. So cytochromes are quite important to distinguish them. So in gamma metanotrophs, those electrons that drive methane oxidation are coming from methanol dehydrogenase. Methanol dehydrogenase oxidized methane takes electron from methanol, takes electron from methanol and then gives those electrons to drive methane oxidation by PMM. Some organisms can actually do direct delivery of electrons to cytochrome C. That's called direct coupling. Other organisms, and this is all in mechanotrophic field, other organisms are doing the electron delivery through complex 3, which is cytochrome oxidase, oxidoreductase, and then through quinone pool, which is complex 2, and that's called uphill transfer. So usually direct coupling and uphill transfer belong to the same category. So the common between them is that electrons are derived from methanol dehydrogenase when it oxidizes methanol to formaldehyde. Alpha-proteobacterial metanotrophs, and this was a big mystery a few years ago, are taking those, so electrons that drive methane oxidation in alpha-proteobacterial metanotrophs do not come from methanol dehydrogenase, they're coming from a quinol pool, like complex 2. And these electrons, so quinol is in complex 2, but electrons that came to quinone and made quinol are coming from complex 1, which is NADH dehydrogenase. So this was actually, and then from that quinol pool, electrons that originally come from complex 1 are delivered to MMO. So basically complex 1 and 2 are like we know are in giving those electrons, unlike in gammas when it's associated with cytochromes and to methanol dehydrogenase. Now, we would think metanotrophs, because they have that very same enzyme like ammonia oxidizing bacteria, they have methane monoxidase. So they could technically oxidize ammonia because same enzymes, methane, ammonia, similar structure, so they could recognize it. So, but metanotrophs, it was found that they can use actually nitrogen as a nitrogen source only, not to get energy from it, just as a nitrogen source. So the big question was, how is that possible? Like even if they can oxidize this ammonia via methane monooxygenase, because it's the same enzyme like ammonia monooxygenase, the product is toxic. It's hydroxylamine. It's toxic. How they do it? So it was found that they are able to use ammonia as a nitrogen source. They have a metabolism to detoxify hydroxylamine. But one weird thing was that they could use ammonia or ammonium as nitrogen source, but not as energy source. Like, why they cannot just extract electrons from ammonia and just send it to cytochrome, to send it to electron transport chain, run electron transport chain, generate proton motor force? What's wrong? Like, they have an enzyme, they have methane monooxygenase. It's the same like ammonia monooxygenase. So that's literally the same, like that's what ammonia oxidizers do, right? But the problem is that this is what people found, the difference, because they try to grow metanotrophs on ammonia only, thinking that, well, why not? They have energy. Some of them can fix carbon dioxide and give them that as a carbon source, and all good. They can't. Why they can't? Because when you compare that ammonia oxidizers have what mechanical stones, so it was found that ammonia oxidizers actually have put additional cytochrome, C552 and 554, and those two cytochrome actually stick those electrons from ammonia oxidation. They could stick those electrons, and then those cytochromes are actually delivering those electrons. So two electrons go to ammonia oxidation, but two of them will be delivered to electron transport chain, and that will contribute to proton multiple generation. Metallotrophs don't have those two cytokines, so they cannot actually use electrons from ammonia and hydroxyl oxidation because there are no cytochromes to carry the extracted electrons, so they can use it as a nitrogen source, but not as an energy source. And again, that's why this is a figure from paper. This is not testable, so don't worry about that. I'm just showing you so you can see how complicated is nitrogen metabolism in ammonia oxidizers and in metallotrophs and generally in all metabolisms. It's way more complicated than the nitrogen. And I'm very willing to discuss that with you. Now, another thing that it's going to be interesting for testing is how carbon gets assimilated in those methyl oxidizing bacteria. So remember we talked about that labeling, when you label glucose at position 1 and when you label glucose at position 3. So the reason why I didn't take this out from lectures, even though I never really asked that, it used to be part of problem sets that was given to students to play with, like for marks, like homework. I thought it's interesting for students to know that C13 labeling and how can you distinguish different pathways. So remember when we talked about how do we know whether bacterium is using EMP or EDP pathway based on which position you see labeled carbon. Same is here, just that it's much easier in methanotrophic world because we don't, unlike with glucose where you can put a label on 6 carbon atoms, we can put it only on 1 carbon. So, and again, just because it might be useful especially for some grad students who are working in this field. So I might want grad students to know this, but not undergrads, although I will tell you. So, unusual carbon oxidation. So, how you do metabolic track. So, how would you know? So people who did this analysis were wondering about methylophenol, methylfocus alkylphenol, I think I called it just 20. This is methanol 20, very famous in biotechnology. Why they have to know how the carbon is assimilated, because they have to know where to delete or add gene, because you don't see where the carbon was. So they didn't know whether this organ is using impurity pathways. So yes, it's using the blue organ monophosphate, but pathways are similar to another. But what after that? It's impurity. So they labeled methane with radioactive carbon, and you can see, so the red stands for label. So this is how it would go. So you would see the straightforward pathway, you would see, but what happens now? So if it's using EDD variant, but you will get your phosphate, you will see carbon 1 5 later. Because it will go to this. But if it's using EMP, you will see carbon in position 3. So again, the purpose of me giving you this detail is just so you understand one that we talked about last week. We talked about why you see sometimes a filter quantity, sometimes CO2 label depending on whether it's EMP or EDD or you label glucose. So for me, this was easier to understand because you literally track the process of metabolites. You have to know these metabolites obviously. But when you look at the structure, you can see how they know? So they track it, and at the end of when they grow bacteria. And at the end of growth, they extract all metabolites and then they search for fiber and they see on which position fiber gets labeled. If they in position with fiber three carbon molecules and the one labeled one. So if they see that carbon in fiber in position one is labeled, that means microorganism is. If they see fiber in position three labeled, that means microbe is using PNP pattern because depending on which variant they use, they perform different metabolic formations, different metabolic formations, and they can and you will see fiber labeled in different positions. Again, we talked about this when we talk about glucose, but glucose might be a little complicated. This is way easier to understand. And we came to the end of Unit 6, which is a brief summary. What do you need to know? Well, obviously, you don't have to know the whole enzymes of fiber. Even I forget. But these are three enzymes that are landmarks. Key metabolize 1,6-fructose bisphosphate or FBP. That's going to be easy to memorize. EDP variants of glycolysis, KDP aldolase, and 6-phosphogluconate dehydratase. I usually insist on KDP aldolase because that KDP aldolase is real indicator, and the KDP and abbreviation is all I ask you to know. I don't want you to know all the name. Pentose phosphate pathway key metabolite 7-phosphate, and I'm not going to ask any enzyme from that cycle, but cellulose is a big indicator of very active pentose phosphate cycle. TCA cycle, obviously alpha ketoglutarate and fumarate and succinate, but alpha ketoglutarate the most important and the enzyme that acts on alpha ketoglutarate dehydrogenase and fumarate. And for glyoxalate cycle will be malate synthase, it will be malate synthase and isocitrate lyase, and key metabolite is glyoxalate. And that would be for this lecture. Don't run away because we are about to move to the next unit. Any questions? I'm listening. I'm just searching for another lecture. I did not share slide. Sorry.
You don't need to know like total numbers of it.
No, God, no. What, like, you see, even though I sometimes insist on knowledge, I don't think it's useful for you to memorize something that you will forget the first moment when you finish this class. And